out there, or afternoon or evening, depending on where you are. Hello, my name is Dr. Rita Blake. I am the Manager of Education here at the California Nanosystems Institute at UCLA. So for those of you who are here with us today for the first time, welcome to CNSI and welcome to UCLA. I'm going to be the MC for today for these presentations. And first of all, I wanted to give you guys a little bit of an introduction to what CNSI is and to what your students have been doing for the last 4.5 days with us. So the CNSI is a pretty interesting place. Unlike many of the other departments on campus, we are actually not a typical academic department. We are a research institution that brings together public and private investment to look at the issues and the research questions that are centered around nanoscience and nanotechnology, which is science and technology at the nanoscale. And if you don't know what that is, I'm pretty sure that by the end of today, you will. So basically, we have 4 different major thrusts that we focus on here as an institute. One is team science, which means that we bring together faculty from all over the campus in science, in technology and engineering and math to do research, again, around nanoscience. We have a really big set of technology centers downstairs that many of which your students actually got to work with for this course, especially our microscopes, which you guys will learn about. We have an entrepreneurship program where we have an incubator that houses around 20 or so startup companies, not exclusively in nanoscience and nanotech, though many of them do enter that field. And of course, we have an education platform, one big component of which are these summer programs. So there are a lot of people, both in the Institute and who worked in this program, that helped make today possible and this whole week possible. So I need to do some thanks before we get into everything. So first of all, our director, Dr. Jeff Miller, who is also actually one of the guest lecturers for the students because he does amazing research on the microbiome, on precision antibiotics, and so he talked about his work. And our executive director, Sonia Luna, who helps everything run in this building and help support all of our activities. We had a lot of staff working both with students and behind the scenes. So I want to specifically thank these 4 people here, especially Dr. Elaine Morita, who is our education program coordinator and who helps keep all of the chaos that is involved in running a summer program together. So I have to give a big thank you to her. Thank you to our events coordinator, Jeremy, to our events manager, Kevin, who helped get everyone prepared this morning and was helping us throughout the week, and also Mark, who is in the back as well, helping with live streaming, and who put together the wonderful slideshow that is out in the lobby of the things that were happening during the week. And then in terms of the instruction, we have had, again, multiple people assisting. We had 3 main instructors for this course, one of whom is sitting right there, Dr. Adam Stieg, who is the Associate Director of the Technology Centers and also the Director specifically of one of the centers, the NanoPico Characterization Lab, and the Technical Interim Director of the Clean Room. We have Dr. Laurent Bentalila, who is the director of the Advanced Light Microscopy Lab and also a researcher here at the CNSI. So he worked with the students on light microscopy. And we have Dr. Hong Zhou, whose focus is on electron microscopy, and he runs the Electron Imaging Center, again, one of the tech centers here at UCLA. So those were their 3 main instructors who each gave them instruction on their specific technique of expertise. We also had major help from Dr. Michael Lake, who is an assistant instructor for this course, and he is a staff scientist here at CNSI who is familiar with all of the techniques as well. He was there to support the different labs that they were doing and also give them feedback and advice for the projects, so kind of serving as an advisor for them. And we also had a wonderful teaching assistant this year named Brian Zhang. He is a graduate student here at UCLA, and he was also there to help them out in the evenings with homework, with projects, and it was really, really wonderful to have him this year. So I have to thank all of the instructors for all of their work this year. So it's been a very, very busy week for these students. They've been doing quite a lot in a very short period of time. In the 4 days that they've been with us thus far, today being the 5th, They've learned about 3 different advanced imaging techniques so that they can see things on the micro and especially the nanoscale. They learned about advanced light microscopy, they learned about electron microscopy, they learned about scanning probe microscopy, and they learned about these 3 techniques with the goal of understanding how these imaging tools can be used to actually ask and answer questions and to develop new cures for diseases, new strategies for discovering new technologies. And specifically, we asked them to look at these different techniques in the context of 2 different grand challenges, which are the Brain Initiative, which looks at understanding the brain and developing new tools that are inspired by the brain, and also the microbiome, which they will, again, I am very sure in these presentations, tell you about. So for these final presentations, we asked them to do one of 2 things, either to tell us how can current instruments address a question of their choosing within these 2 grand challenges, or what new instruments should be developed to address one of these grand challenges. So they put themselves into groups of 3. They're going to present now. So each group is going to have between 6 and 7 minutes, roughly, to do their presentation. I will be timing, so at the 7-minute mark, you will hear a noise, which doesn't mean I'm going to pull you off the stage, but it means to try and, to wrap it up if you haven't finished yet. But they'll get around 7 minutes. And then we'll have about 3 minutes of Q&A. And by Q&A, this is open to everyone. So students, if you want to ask questions, instructors, of course, will be asking questions. Parents, if you guys have questions, please ask them. This is your opportunity to learn about what your students learned. So please, please, please ask questions. It is a more fun time the more questions get asked. So please do not be shy about asking if you don't understand something. So with that being said, I am going to transition it over to our students. So team 1, would you please come up? And let's give all of our teams a round of applause.
Good morning, everyone. So today our group will be talking about the gut microbiome and its relations to depression. My name is Spencer Gout. These are my colleagues Alex Meyer and Emma Scaff. All right, so let's get started. So over the course of the week, we've been addressing a few different types of imaging techniques. But more importantly, we have looked into how they can be applied to real life. So our group today, we took interest to how microscopy can be used in the context of medicine. And today, the medical challenge at hand is a serious and worldwide disease that affects many. So we will be addressing the broader challenge of the microbiome using microscopy. So currently in the gut, there's a family of bacteria called Lactospiraceae. Lactospiraceae are known to have a positive correlation with major depressive disorder, otherwise known as depression in humans. So this correlation is known, however, the causation isn't known. The functions of the bacteria, so we don't know this, and that's what's being left out in the open for us to figure out. So whether this is on the chemical basis of a neurotransmitter being inhibited on like a neural synapse, we really don't know yet. So that's what we're trying to figure out. So in our experiment, we plan to use fluorescence microscopy to monitor the bacteria and the relationship with neurons in the gut, so to further understand our gut's microbiome and how it affects our central nervous system.
Wait. Do you just want to hold this?
Yeah.
Sorry.
So we will use this information to determine if neurons and their relationships with bacteria elicit depression itself. So we chose fluorescence microscopy. So for this project, we chose fluorescence microscopy especially. And I think Dr. Bentolila said it best when he said that the main goal of fluorescence microscopy is to derive an image based on the interaction of light and matter. So this is done by passing forms of light, say a laser, and photons being passed through a series of lenses and mirrors until a proper image is produced. And with this image, researchers can analyze in further studies and conclusions. Now, there are two major advantages to using fluorescence microscopy in the context of our experiment. One is that this technique is non-invasive and will not damage our cell cultures. And secondly, its resolution is strong enough that it will be easily able to see the neuron functions. So as I said earlier, fluorescence microscopy can be used in the gut microbiome. So pictured to the right is a spatial organization of various gut bacteria. And pictured to the left is a three-dimension landscape of the gut's microbiome in general. And these were taken with fluorescent microscopy techniques. So I'll pass this on to Alex, who will talk about the details of our experiment.
Okay, so with what we currently know now, there are several groups of bacteria that reside within the gut microbiome that, that relate to depression. Sorry. But what we don't know, like Spencer said, is that we don't really know like the importance of which ones cause it, which ones have like a greater effect at causing depression, and we don't quite know the specific functions and how each of the bacteria like cause depression. But despite knowing like there are a whole bunch of these different types of bacteria, we're specifically focusing on the bacteria in the gut microbiome that fall under the Lactospiraceae family. Due to research that has been previously done, we are aware that there is a positive correlation between having these types of bacteria in your gut microbiome and the chances that a person would experience the symptoms of depression. We currently do not know like what mechanisms allow for this to happen though, and that is what our study is trying to figure out. So specifically, our study is trying to figure out if the mechanisms that cause depression are due to direct relationships between bacteria interacting with the neurons in human guts. So to run this study, we are going to first grow neuron cells and cell cultures. Then we are going to introduce bacteria that I've previously mentioned. But in order to be able to see anything going on, we need to encode these bacteria with genetically encoded calcium indicators, or GECIs. Just like how fish can be encoded into bacteria and works as a fluorescent marker for proteins, GECI can also be encoded into bacteria and works as a fluorescent marker of calcium 2 ions, which would help us see if there is a relationship between the bacteria and the neurons. And the use of light microscopes allow us to see this happen in real time. And I will let my colleague Emma explain the importance of what our findings may be.
Depression is a significant problem in the United States, seeing as it affects 7.1% of the entire population each year. Through determining the gut microbiome's relationship with depression, solutions can be discovered in order to hopefully reduce the amount of affected Americans, as well as people worldwide. From the setup of our experiment, the relationship between the gut microbiome and depression, we can deduce that there will be one of two outcomes. The first case scenario would be that the results show a definitive connection between the microbiome and the disease. Through the results involving calcium ion channels, we will be able to see the bacteria being used interacts with the neurons in the cell culture. Thus, the connection from the gut to depression will be proven. Therefore, a reliable clinical treatment can be found based on how the microbiome proves to affect the body. The second of the two outcomes is that there will be no feasible correlation between the bacteria in the microbiome and depression. These ambiguous results will be shown by the bacteria simply not having any interactions with the neurons, proving the lack of connections between the two factors. If the experiment does not display the predicted effect, the outcome will still be able to contribute to the research field of the causes of depression. By ruling out the possibility of bacteria from microbiomes interacting with neurons, focus can be pointed elsewhere, putting researchers closer to the end goal of finding the gut microbiome's overall effect on the body. No matter what the final outcome of the experiment is, it will undoubtedly help to progress the research community, whether it be by finding a new connection to assist in finding treatment or permanently ruling out something that was once thought to be a cause of depression. The data found will be able to, in some way, further research surrounding the gut microbiome. have anything after this?
So during the Q&A, if you guys could please repeat the questions because they are not mic'd. So for the people who are watching, it's helpful if you can say the questions. So
Any questions? Yes.
First of all, congratulations. A very nice presentation. It's the first one, so I know it's easy to be the first. My question is to you guys, like, from what you have been reading, you mentioned like a positive correlation between that specific bacterial type and depression. And my question is, do you know if that specific bacteria is present normally in the human microbiome, or it's just not present at all, and this is when you have it, but you show signs of depression?
That's a great question.
Repeat the question.
Oh, yes. So is the bacteria that we're looking into, is it found in like humans without depression, or is it like just found in reduced amounts? From our research, I'm pretty sure it's just that there's like an abnormal amount of it, like there's an increased amount of that type of bacteria in human gut microbiomes. But yes, sorry.
As a follow-up to that, how would that help you decide what to do about that? For example, would you want to, for example, kill all of those bacteria?
So the question was, how would that, like, how can we take understanding if this is present in all people, how can we, like, create a treatment for that? Any of you have an idea of this?
So I think if there's like if a connection is found, say because serotonin is a neurotransmitter correlated with happiness in the body, so a lot of what depression does is inhibit serotonin from binding onto the neural synapse. So if a relationship was found with the neurons and the bacteria, then we could possibly deduce with further research that the bacteria are likely producing some sort of substance that attaches onto that receptor, onto the neural synapse. So what could be done with clinical treatment is to target, using antibodies or some form of that, is to target what is being inhibited on that receptor site. So that's one thing that we could do about it.
Can I say one more thing on that? I don't think completely killing off that bacteria may be entirely beneficial, though, because like a regular biome with animals, there are specific niches that each of these bacteria fill within you. And I think if you kill off all of one type of bacteria, another harmful bacteria is allowed to come in and replace it. It's like what's happening when you take antibiotics. You're killing off all the bacteria, thus allowing for much deadlier things to come in and affect you worse. Yes.
Another question. I really like the way you break down your experiment, right? Trying to really outline how you would do the experiment. So you explain how you would mix the bacteria with a culture of neurons and how you will use the optical microscopes to look at them. So my question is, do you have any idea of what you would be looking for?
To just start off, I think we would want to be looking for like just an indication that like the bacteria is sending out calcium ions in some sort to like activate the neurons. But if we find that relationship, then more can be done to look into like what specifically is going on with the bacteria, because if they're not doing that, then there's no point to like look for more. So this is just the starting point, and more research can be done after that.
And so to add on to what he said, I think more specifically what we'd be looking for is like a presence of the lacnospiraceae in between the synapses, right, in between the neurons. Because like I said, serotonin, and I'm sure there's other neurotransmitters that are correlated to depression, but the basis of this would be looking for specific inhibitory responses to serotonin. So we'd be looking for a presence of that between those neurons.
see if they're in one particular sensitive location of the nerve.
Yeah, and that could be, motor neurons, inner neurons, or sensory neurons, but further research should be done.
Oh.
Yeah.
How are you planning to use calcium 2 plus to assist in your research again? Like you mentioned that you would coat bacteria with calcium 2 plus.
What? Oh, no, we're not going to code it. Oh, sorry. How are we going to use calcium 2 ions to help in our research? You said that we are going to code it. No, we are going to encode, sorry. We are going to encode things called genetically encoded calcium indicators, which basically attach fluorescent proteins to calcium ions produced by bacteria so that you can simply like track them. And neurons work by having calcium ion gates that like start transduction. So we're just looking for that at first, if that answers your question. Okay.
All right. Thank you, guys.
All right, team 2. This is you guys, right?
Ready? Ready? You should take this. 3, 2, 1, go.
Okay, hello everyone.
Hi.
So we did our project on multiple sclerosis mediation. And the way we're going to do that is by analyzing T-cell interactions via electron, light, and scanning probe microscopy. My name is Giovanni Vignone.
My name is Wanjio.
And I'm Rashid Chalapa.
So today our question would be, how can electron light and scanning probe microscopy be used to understand the interaction between Treg cells and cytotoxic T cells in patients with multiple sclerosis?
And can we use this information to reestablish the boundary of the immune system and to mediate or prevent further degeneration of myelin in the neurons?
So here's our abstract. The autoimmune disease multiple sclerosis causes degradation of the myelin sheath in neurons, possibly due to the deregulation of cytotoxic T cells, which ultimately results in progressive reduction of muscle function. After an analysis of the roles Treg cells play in the immune response, we propose multiple methods to mediate or even stop MS. Considering that CD4 positive, CD25 positive Treg cells can suppress the immune response, through imaging the way that these receptors actually interact with through various microscopes modalities, we can study the dysfunctional mechanism and develop a strategy to restore the integrity of these cells.
So just some brief background over multiple sclerosis. So this autoimmune disease involves the attack of myelin, which protects nerve fibers, and deterioration of connections in the central nervous system, causing a dysfunction of the peripheral nervous system. So eventually this results in permanent damage to the nervous system itself, loss of motor function or paralysis, depression, and epilepsy.
In addition to that, T cells are a type of lymphocytes, which is a white blood cell from the lymphatic system that are produced in the thymus, which is a gland between the lungs. T cells also play a central role in the immune system and mainly distinguished by other cells through their glycoproteins such as CD25 positive. In addition, cytotoxic T cells are a specific type of T cells that are used to eliminate virus infected cells, tumor cells, and other foreign invasive cells. Not only that, Treg cells are a specific type of cells. They actually monitor the immune response, specifically Treg cells with CD4 positive and CD25 positive receptors are in charge of stopping cytotoxic T cells from destroying other cells. Finally, T cells turn into Treg cells by going through differentiation through to transcription and translation of FOXP3 genes.
So for the current situation, through successfully performing this experiment, we could mediate or even halt progression of multiple sclerosis, which affects nearly 1 million people in the United States alone. So this number has doubled since the previous study, which was conducted around 3 years ago.
So some precedents that has been worked on by other scientists would be first Alla and Hens in 2008 found that due to the deletion of FOXP3 in mice, which led to the reduced expression of putative suppressor genes, which is a characteristic of Treg cells, and it suggested that with FOXP3, Treg cells are able to produce faster and more efficiently. In addition to that, researcher Alla and Hens also found that mice who had EAE,
which is experimental autoimmune encephalomyelitis.
The MS version for mice, were able to prevent getting EAE after receiving Treg cells from the wild mice and the mice recovering from EAE. And finally, the suppressive ability of CD25 cells, CD25 positive cells were derived from CD25 negative cells in vitro proliferation assay. The CD25 positive cells were energic and suppressive, but the CD25 negative cells, from which the CD25 positive cells were derived from, proved little suppressor activity in autoimmune diseases.
Okay, so here's our project proposal. So in order to truly mediate or even halt the progression of multiple sclerosis, we must first identify the origin of the autoimmune disease. So we believe that at least for some cases of MS, this is due to T-cell interactions. So for our proposal, we're going to begin by drawing a blood sample from the lymphatic fluid in a patient with MS and one without MS, the one without acting as the control. We will then sort the cells via flow cytometry, which is a technique used to sort cells, and those cells will be Treg cells and cytotoxic T cells. Then we will compare the amount of Treg cells in a normal patient to the one with MS. And if the Treg cells are low or not present at all in the patient with MS, we think that this would be due to the FOXP3 gene, which we talked about earlier, not being expressed. That FOXP3 gene codes for proteins, which are used for the differentiation of T cells to Treg cells with CD25 positive receptors. And the way we'll tell whether this gene is being expressed is through Western blotting, which I don't have time to go into. If this is low, if the gene expression is low and there's no proteins, there's low amounts of proteins found in patients with MS, we thought we could be able to actually introduce the FOXP3 proteins into the patient with MS, or even do a blood transfusion with Treg cells. And if we do find that there's normal amounts of Treg cells, we believe that the issue could be actually with a mutation on the receptors between the Treg cells and the cytotoxic T cells. Yeah.
So for this experiment, we'll be applying the microscopic techniques we learned over the past few days, which are the cryotransmission electron microscopy, or cryo-TEM, atomic force microscopy, and confocal microscopy. So for cryo-TEM, electrons that have very small wavelengths are fired at a sample, allowing us to scan very thin segments of the organism in order to produce black and white two-dimensional images of the internal structure. So we may image the bond of the receptors between the cytotoxic and the T regulatory cells in patients who have multiple sclerosis and those who are healthy, to determine what constitutes the difference between the two types of patients, which could be a mutation. And for atomic force microscopy, we could analyze the repulsions from the electron cloud in atoms on the surface of the sample by using a thin tip probe, which is made up of like one atom, and monitoring a cantilever. We'll attach a CD25 plus T regulatory cell to a tether attached to a probe and measure the affinity between that cell and the cytotoxic T cells. And finally, for confocal microscopy, we'll produce a 3D image of T cell interactions.
Okay, and on to contributions. So from the experiments and the ideas we had, we will be able to give the science field a potential solution for MS. Another is we will approach this problem we would give this problem a different approach, which is that ever since from the start, scientists have been trying to implant T cells into EAE diagnosed mice, and we're trying a different approach where instead of injecting more Treg cells right away, we would determine the level of it first to see what is the right approach to the patient.
So our experiment is addressing those patients with multiple sclerosis using only T cells. So a challenge would be aiding the patients who have multiple sclerosis, but it's not caused by the Treg cells and they do not have mutations. So we may also encounter issues freezing the Treg cells and cytotoxic T cells with when their receptors interact, and this crystal structure may also distort the natural form of the interaction when we freeze it.
Okay, so as for the future direction, we need to actually perform the experiment to find what is the defective link between the T cell interactions, and we also need to, if that is the case, and there is an issue with the T cell interactions, then we can actually help patients with this this issue with MS. And if that's not the case, then maybe look at the other causes, like is there a defect in dendritic cells presenting antigens of myelin. Okay, and here are our references, and that's it.
And please don't forget to repeat the question.
Okay.
Does the advent or the newness of the technology in this microscopy lead to you guys and everybody, I guess, you're able to attack these new perspectives on these diseases, afflictions? And this is a layman's question, but that's what I'm getting from this, is that we're at this level of technology that you're able to now dial into these specific things. Would that be correct?
So his question was, are new technologies actually allowing us to observe these interactions and the issues causing MS? So as we talked about, we were talking about using scan probe microscopy, which is a relatively new type of microscopy, and we were actually talking about attaching a Treg cell on the edge of that probe and measuring the affinity of the receptor junction between that cell and a cytotoxic T cell, which is, I think, a relatively new type of project. Yeah, so that would be where that comes into play.
Okay, I actually do have a question. So in a little schematic you showed the U.S., but it was split in half with the North and the South, like kind of thing. But there were exponentially more cases of MS in the North. Do you have a reason why that might be?
She's asking whether the North, or why the North would have more MS. I'm actually not too sure. The only thing that I can think about is that it has to do with the environment, and something's causing issues with the immune system, or like it's causing defects with the immune system in the North. That's maybe different to the South.
And elaborating on that idea from the trends we studied, essentially, like, since much of the northern United States lie on the same latitudes as Europe, the northern part of the world, Europe, North America, these areas have a substantially higher risk of multiple sclerosis.
Gentlemen, this is very interesting. Clearly, this is a huge impact on people, over 1 million people. Is it possible that the differential between the hemispheres is perhaps population based? But the question I have for you is are you saying by looking at the actual junction that where the Tregs join onto the nerves, that by looking at that, that is how you do assume that is where the problem of the breakdown of the myelin is by looking at that you can actually tell that from the microscope?
Okay, so his question was whether looking at like the actual junction between the T cells is the reason for multiple sclerosis and causing cytotoxic T cells to degrade the myelin. I think that's what you're asking. So, well, multiple sclerosis there's no cause for it, and that's likely due to there being many reasons for this issue. But we kind of are trying to narrow that down to maybe a specific set of cases where there are T cell, there's issues with T cell interactions. So we were thinking we would determine if, so Treg cells calm down the immune response. So we were seeing if there was an issue with those Treg cells calming down the immune response, whether there's low levels or so they can't calm down the immune response. And the junction between the Treg cells and the cytotoxic T cells, it calms the cytotoxic T cells down. So we were thinking that's maybe our solution. And the issue with multiple sclerosis is that the cytotoxic T cells degrade the myelin on the neurons. So maybe they're like, they have too much of a response, and they're doing that like really frequently, and that's causing multiple sclerosis. And maybe this could be like the location or the origin of the issue for some patients.
So not really a question, more of a comment. This is great. Like, hypothesis-driven project, right? You have an idea. You've very thoughtfully designed experiments using multiple modalities. Both you learned about in this class and outside of it, right? To explore a question that is unanswered, right? I know that obviously you have lots of references. There must be some, I would assume, indication that these interactions are implicated in this disease at some level. But I just want to say, in terms of all the groups, like this is the type of project and the type of rigor that we like to see, right? A lot of it is not just saying we are going to use a microscope to look at this, but the question is why? What is the question that you're trying to answer, right? And that hypothesis you have about these interactions between these two types of T cells is a really inspiring thing to see from a group of young people, right? To come up with an idea like that, whether it's inspired from the literature or not, but then really delving deeply into it. I just want to say great job to this group. I'm sure we'll see great ideas from everybody else, but for all the groups that follow, right, they did a fantastic job using hypothesis-driven science, right, as a mechanism for discovery. So I just want to say really good job to you guys. And
Thank you so much.
Thank you.
Someone else do it. I can't. I'm shaking. Okay, fine.
Hi, I'm Maddie.
I'm Mia. And I'm Neha.
And so our project is about the gut microbiome's effect on autism.
So autism is a mental disorder and is identified by a spectrum of difficulties, including social and communication difficulties. It can be developed through genetic predisposition or through environmental aspects that affect the gut microbiome. An unhealthy gut microbiome affects the development of autism by causing inflammation in the body that travels to the brain and can affect the development of autism. Lisa Azizzadeh, along with scientists from UCLA, were some of the first to research the connection between the gut microbiome and the brain. Since the discovery of this connection, some treatments have been developed, such as a fecal transplant, where a healthy fecal sample is transplanted into the digestive tract of a person with autism in hopes of creating a healthier gut microbiome. This transplant takes about 2 years to show results.
The gut microbiome is a group of millions of microorganisms that reside in the stomach and the intestines. It is a rapidly changing environment of bacteria that is very vital in bodily health and regulation of disease. Though there are many trillions of microorganisms in and on humans as a whole, the gut microbiome in particular directly correlates to the different functions of the immune system. A balanced system ranges from controlling digestion all the way to managing allergic reactions, while an unbalanced system can cause problems including obesity, diabetes, and more recently, autism. Through recent discovery, it has been proven that kids with ASD have different gut compositions than those without.
A recent study this year done by UCLA and USC scientists proves that gut bacteria activates the central nervous system to send information to and from the brain. Additionally, knowing that the gut is largely affected in diet, new studies have emerged to restrict what children diagnosed with ASD digest. Yet these studies prove to be inconclusive because not all severities of autism can be lessened by a change in diet. However, by analyzing the fecal transplant bacteria properties held in stool samples, scientists were able to come up with a connection between said properties to known behaviors in children diagnosed with autism.
During the 1800s, a scientist from Boston, Massachusetts began recognizing autism as a mental disorder. Scientists were interested in how people with autism were challenged in some areas, but at the same time extremely talented. At this time, scientists were not 100% sure of what autism was. The word hadn't even been invented yet. This new discovery sparked scientists and researchers' interest in studying this newfound disorder. During these early stages, scientists and researchers diagnosed autism by watching and analyzing the behavioral patterns of an individual. It was not yet known that the gut microbiome had anything to do with autism. Scientists continue to study autism to this day, but at first had very limited knowledge on the subject.
The discovery we found was that the microbiota transfer therapy, commonly known as a fecal transplant, were widely successful between those who have a purely healthy gut microbiome to those children who possess an unbalanced microbiome due to being diagnosed with autism. A recent 2019 study by Arizona State University concluded that 50% of children who were diagnosed with autism, that autism symptoms reduced nearly 50% 2 years after the fecal transplant. The way it works is relatively simple. As the gut microbiome of a healthy child has stronger and more valuable bacteria, if you take the specimen from the feces and transplant it into the digestive tract of a child diagnosed with ASD, the hope is that the bacteria would create a healthier environment in the gut so that they can balance their microbiome over time, which proved to be very successful.
So, autism can either be caused by a genetic predisposition or it can develop through environmental influences that affect the gut microbiome. An imbalance in the gut microbiome can cause inflammation in the body, which can travel to the brain and may result in the development of autism. Lisa Aziz-Zadeh from the USC Division of Occupational Science and Occupational Therapy stated, "If you ask the parent of a child who has autism, there's a good chance you will hear stories of tummy troubles like constipation, cramping, and pain." In this quote, Aziz-Zadeh highlights the external signs of the internal imbalance in the gut microbiome. The gut microbiome is responsible for many important bodily functions such as immune system and metabolic functions, regulating disease, and vitamin production. A fecal transplant is a newly developed treatment that has shown promising results. In a study by ASU, as was just stated, they found that 2 years after the transplant, 50% of the patients decreased their symptoms by nearly 50%. This discovery is just one step further into the future of treatments for autism.
So our ideas for the future is we will be using an optical light microscope to study the detailed structure of the bacteria in a healthy gut microbe versus an unhealthy gut microbiome. Our plan is to improve the mechanisms of the fecal transplant by minimizing the amount of time that it takes to reduce the severity of autism symptoms. As previously mentioned, the microbiota transfer therapy takes about 2 years to see its full effect on ASD symptoms. For people that are higher up on the spectrum, this is 2 years of life that they have to live before change starts to occur. Our action plan is to infuse the species of the feces before the transplant with naturally good bacteria such as raw probiotic strains. Probiotics are live bacteria and yeast that are generally very good for you, and when ingested, they can take residence in the gut and naturally improve your health. By infusing the species with probiotic strains, the goal is to increase the rate of growth for the amount of healthy bacteria to keep the microbiome sustained. Of course, further research has to be done to see if the feces would react accordingly with the probiotic strains.
Contributions. One major contribution was the discovery of the fecal transplant. Stated by the medical press, fecal transplants help to substantially improve behavior and gastrointestinal health in children with neurodevelopmental disorder. These transplants are not used from pure fecal matter, but instead purified matter that has been extracted from feces. Fecal transplants rebalance the intestine, which directly influences the brain. Introducing this new bacteria diversifies the bacterial community, also stated by the medical press. After the transplant, and now a new abundance of beneficial bacterial species was formed. The fecal transplant has majorly contributed to helping kids with brain disorders like autism.
And these are our references.
And please remember to repeat the question. Okay, so oh, there we go, thanks. Okay, now you don't have to repeat it because they can hear me. Okay, so I wanted to clarify what exactly you would be looking at with the microscopy technique that you mentioned. So you said optical microscope, and you mentioned wanting to look at the detailed structure of the bacteria. So what structural features are you talking about specifically, and how big are those features? So I guess that's connected to saying, is optical microscopy actually the technique that you want to use to see what it is you're looking for?
So I think what we would do is we would inject GFP, green fluorescence protein, into a stool sample with an active gut microbe in a healthy sample. And with the probiotic strains that we would inject into them, we would see if the probiotic strains would help increase the amount of bacteria that is created when we put in the fecal transplant into the digestive tract of a kid with ASD.
And also I wanted to add to that, so the reason that we chose light is because we wanted, if we were to use the electron microscope, we wouldn't be able to use a live sample, and all the microorganisms, even though they're tiny, they're really tiny inside the microbiome, and we wouldn't be able to use a live sample of that and see everything that's happening inside of there. So that's why we chose the optical microscopy technique.
I truly find that fascinating that you've taken a really clinical problem and are trying to explore what the cause of it is. I mean, fecal transplant is a clinical issue. Autism is a spectrum, right? So it's a spectrum of disease. Have you read anything about the fact that perhaps there'll be different gut flora or microbiome changes depending on what spectrum of the autistic spectrum one would be on? So you're taking a clinical problem and you're trying to find a microbiological cause, which I think is fascinating, actually.
I can answer this one. So wait, I don't need to repeat it, right? Yeah. Okay. So yes, everybody's microbiome is going to be different. What the fecal transplant is trying to accomplish is just introducing more and healthier microbiome into someone with autism who may have an imbalance in their microbiome. So everybody's microbiome is different, but the fecal transplant can help to fill in those imbalances.
I can talk loud. So, in your proposal to utilize optical techniques, do you see that what you learned and saw this week is actually capable of pulling off the measurement that you want? And what I'm getting at is that the optical microscope can certainly visualize bacterial cells. It can image, over large areas, living organisms. It can measure dynamic processes. But did the microscopes that you saw today or this week indicate to you that we're ready to try to do this measurement inside of a live human, for example? Because we talked a lot about how you actually gain access to your sample, right? And the reality at some level is that the gut microbiome is inside of my intestinal tract, how do you get the measurement that you want to do to operate in that environment? Or do you plan to prepare your sample in some other way? If so, what are the challenges with doing that?
I can answer this question. So we read and researched that a lot of studies have been done on mice, because mice and humans have a similar digestive tract and gut microbe that we could study potentially for the bacteria. So additionally, like for current procedures, I guess we don't exactly know how the humans would react if we were to look at their bacteria just straight from their gut microbe. That's why a lot of studies have been done on other animals, such as like mice, that we said, if that answers your question.
Yeah, I guess I was asking, are those studies that you looked into done in live mice inside of their intestinal tract, or are they trying to basically extract, purify, and recapitulate the microbiome in a petri dish, for example, right? Because there's a big difference between what you can do in a dish by just mixing together a bunch of bacteria versus what actually happens inside of your body, because it's not just what it's made of, it's also where it is. The spatial distribution matters, right? So I guess I'm just wondering, in your research, did you see that these technologies currently, and it's okay if they're not, right? Because part of this was, what do we need to do, right, to identify new tools and technologies to enable us to do this type of stuff?
I think currently, I'm not sure if the mice were alive. I think they were just taking out samples of the gut microbe so that we could see the bacteria and see how it, enhances after the fecal transplant is put in. So I'm not sure if there's enough research out there yet to do live gut microbe screenings, but I think that's definitely something that could be looked into.
So I could maybe add a little bit to this. One of the current technology that has been developed is actually, as we touch upon very, very lightly, I don't know if you saw that later on by looking at the literature, is to miniaturize microscopes, right? So one of the very important effort here is to take the microscope and bring it to the tip of an endoscope, for example. So you can think about using an endoscope coupled with a microscope to actually look at the microbiome inside a live mice or a live human.
Yeah.
And this is actually probably going to be done very soon.
Yeah, the studies that we saw were very recent. Most of the studies were in April and May of 2019, so they're very new in coming up with their research, so there's definitely going to be more out there soon.
Thank you.
Hello, this is our final project for Nanoscale Microscopy 2019. My name is Artem.
My name is June.
And I'm Jesse.
Our topic is the utilization of bacteria to degrade styrofoam. Styrofoam, a polyester, is one of the biggest problems in pollution, producing 2.3 billion kilograms of waste each year. It has many problems, including being non-biodegradable, being an oil byproduct, and producing CFCs, which degrades our ozone layer. Therefore, with the problem of styrofoam, we want to create a solution. In order to address the problem, we propose to bioengineer an enzyme into a bacteria, which makes it sustainable in the economical and environmental world.
So now we'll talk about two precedents similar to our experiment. The first one is about the Pseudomonas putea bacteria. And this bacteria was found by Kevin O'Connor and his European colleagues. And it is essentially able to consume pure styrene oil, which is a remnant of superheated styrofoam, and it's also a toxic pollutant. So it's all able to do this once you use pyrolysis to the styrofoam. And it's also able to consume 80% of the styrene oil, and also along with other toxicants. But the reason why, although this may seem very effective, the reason why this is not practical is because you have to superheat this styrofoam. So if you have to put that much energy into the process, it's not energy efficient. Our next precedent is about the Audinella saccharinices bacteria, and this was found in Japan near a recycling plant. And this bacteria is able to consume polyethylene terephthalate, also known as PET, and PET is basically the things that make plastic water bottles. And it's able to break it down using the PETase enzyme. Although this experiment was successful, it's not applicable to styrofoam because PET and styrofoam are two different types of plastic.
So basing our proposal off of these two precedent researchers, we propose that we engineer bacteria that could actually degrade styrofoam into harmless materials. So after doing some research, we figured out that mealworms actually created enzymes called Exigobacterium sp strain YT2 enzymes that actually degraded styrofoam and made them into harmless materials. So keeping that in mind, what we propose to do in our experiment is to take the gene that codes for this enzyme, this particular enzyme, and take it out from the genome of the mealworm by using restriction enzymes to the DNA, so we could cut the gene out and isolate the gene. After the gene is isolated, we propose that we use a cycle called a PCR, which stands for polymerase chain reaction cycle, which basically sort of makes a lot copies of the particular gene that we want, so that we could work with these specific genes. So there's 3 steps to the PCR cycle, which the first one is denaturization, which is basically heating up the gene and breaking the double helix bonds. After that is done, there's a second step called primer annealing, which is basically attaching primers to the 2 sides of the DNA. And the third and last step is elongation, which is basically putting DNA bases, attaching them to the primers, and copying the DNA. This cycle is repeated over and over again, and each time the cycle is completed, the number of the gene will exponentially grow.
So after we created all these DNA fragments, we now want to implement it and put it inside the bacteria. So in order to do this, we have to get a plasmid, which is a circular piece of DNA, and we would cut the plasmid using restriction enzymes. And now that since we have a gap within the plasmid, we're able to insert our fragment of DNA into the plasmid, and we seal it all up using DNA ligase.
So with the resulting plasmid, in order to put them into bacteria and actually use these plasmids, we do this technique called the heat shocking, which is basically forcing plasmids to enter the bacteria that we want, and sort of having the bacteria force the bacteria to incorporate the DNA in the plasmid into its genome. So after all that's said and done, with the bacteria, we could culture them in an environment that is styrofoam rich.
So now that we have our culture of bacteria, we now want to see if the bacteria is actually consuming the styrofoam and actually digesting it. So we're able to do this by using the fluorescence microscope. And basically, we're able to do this by first getting the GFP gene from a jellyfish and integrating it into the bacteria's genome. And once we do this, the bacteria will be able to light up. So now that it lights up, we can put it under a fluorescent microscope, and we're able to see if the bacteria is able to thrive or it perishes. The reason why we chose fluorescent microscopy is because compared to the other alternatives, it's much more practical. So for example, if we used electron microscopy, the electrons shooting from the electron microscope would damage our bacteria, which would cause unwanted effects. And if we use, say, scanning probe microscopy, then it wouldn't yield great results because the scanning probe microscopy will only sense for the surface of the bacteria, whereas we want the entire culture.
So during this experiment that we propose, there's one major challenge that we might encounter. So that challenge is basically, even if we incorporate the gene that codes for the enzyme, which is called Exigobacterium sp strain YT2, that actually breaks down the styrofoam into the bacteria that we want, the bacteria still may not be able to create this enzyme because, like, the reason being is because bacteria cells are much less sophisticated than mealworm cells, which the enzyme originated from. Therefore, the bacteria itself may not physically be able to create these enzymes. So one possible way to solve this problem is by sort of researching this particular enzyme in depth. So looking at structure and maybe its critical points that actually does the work of breaking down the styrofoam. And after we identify that, we can maybe modify this enzyme that is compatible with the bacteria and put the enzyme in the bacteria. So if everything goes to plan and the experiment does succeed, one future direction we could take this experiment is by mass producing the resulting bacteria and sort of releasing them into environments around the world that are maybe very styrofoam polluted, and thus cleaning up the environment. In a short summary, we want to take a gene from a mealworm, make an enzyme, and then bioengineer the enzyme into a bacteria. If we are successful, we will be both economically and environmentally sustainable.
All right, questions?
Wow, another great presentation. Thank you. Question: how long do you think, or how long do you think it will take to degrade the styrofoam?
From the research we've done with mealworm enzymes, it is said that it could sort of, in a day, it could degrade, I think, a pill's worth of styrofoam, and so it is still a significant amount if we put a lot of bacteria into that use of degrading the styrofoam. So, so I believe it's not that much, but it is significant enough to sort of degrade enough styrofoam to make a difference.
I want to point out that rather than using, because obviously we can use the mealworm bacteria that already has the enzyme to break it down, but the reason why we're not doing this is that it could potentially become, if we introduce it into new environments, it could become an invasive species and it could throw off balance the general ecosystem.
Very nice.
All right. If there's no more questions,
Yes.
So close.
I wanted to say this was very interesting, and you guys did an awesome job.
Thank
I really
you.
like
Thank you.
learning about it.
Thank you.
This is on or not? Okay, there we go. Okay, so my question is, do you know what the proteins actually degrade this styrofoam into, and like how good is that for the environment, or what would we do with that?
So, sort of clarify your question. Are you asking what protein actually does this work?
No.
Like, what's the byproduct?
What's the byproduct?
Oh, the byproduct of actually this enzyme degrading the styrofoam is, we do still need to do more research on this, but yeah, I can't really tell you right now because we're not really sure.
I think it broke it down into carbon dioxide and other organic molecules. So it's basically generally safe for the environment.
Thank you.
You don't need your computer. Wait, I already have it.
So, hi. Our question is, how can proteins of gut bacteria affect the growth of colon cancer? So, I'm Angela Xiang from Portola High School.
I'm Francesco Donato from Leone XIII High School.
And I'm Harsimer Ball from Moore Park High School.
So, how do certain proteins of gut bacteria affect the growth of colon cancer? Gut bacteria is one of the many types of microorganisms found in gut microbiome of humans. Scientists have found that gut bacteria can promote several host cellular multiplication and survival pathways, contributing to the appearance of colorectal cancer. One issue regarding colorectal cancer is the location of the proteins. It is not clear if the location of the proteins in cancer-causing bacteria affects tumor growth. We propose further studies and experiments on gut bacteria that are associated with tumor growth in the colon in order to determine how they cause cancer. We plan to place healthy colon cells in vitro with sample bacteria and use several microscopy techniques to study how the different bacteria species cause cancerous growth. We conclude that each cancer-causing bacteria will promote colon cancer with different methods.
There are several bacterial populations which directly influence tumor development in the colon. The main three are Escherichia coli, enterotoxigenic Bacteroides fragilis, and Fusobacterium nucleatum. This is relevant because if we are able to compare the movement of the proteins from the bacteria to the colon cells, we will be able to determine how these proteins can cause the proliferation of the colon cells. Answering our research question, we will contribute associated with three main advantages. The first one is that we will have more information about the colon cancer, and thanks to this, the second one is that new treatments can be developed. The third one is that if we can develop those treatments, we can help to cure more people all over the world.
So as of right now, researchers have utilized several microscopy techniques to study and confirm which bacteria in the gut microbiome have the ability to influence cancer formation. Certain bacteria can stimulate cancer formation by blocking immune effectors that normally inhibit tumor growth. For example, F-nucleatum inhibits killer T cells from functioning properly in the immune system and attacking pathogens, therefore helping cancergenesis. The ETBF, also known as enterotoxigenic Bacteroides fragilis, is a bacteria that produces a toxin known as BFT, which directly causes colorectal cancer. Another cancer-causing bacteria, E. coli, produces the toxin colibactin, which mutates DNA in colon cells, and depending on which genes are mutated, the colon cells can grow uncontrollably, resulting in cancer formation. So for our experiment, we plan to observe test tube samples of each of the three cancer-causing bacteria in vitro with healthy colon cells and compare the cancer growth in those tubes with 3 test tubes also in vitro that only contain specific proteins from each of those bacteria. We chose this experiment as it will allow us to determine if the location of the proteins in regards to the bacteria will affect colorectal cancer growth. Our hypothesis is that the protein of each bacteria are the sole cause of cancer's growth, so regardless of if the proteins remain inside the bacteria or are secreted, cancer will still appear. The first part of our experiment is to identify which proteins in each bacteria cause tumor formation. In order to confirm that our samples are of the correct bacteria, we will utilize gene sequencing and compare the entire nucleotide sequence of previously identified samples of each of the cancer-causing bacteria with our new samples. Then, to find which specific proteins in each of the 3 bacteria cause tumor formation, the polymerase chain reaction can be used to cut off all DNA that does not code for cancer-causing proteins and can then amplify the specific protein sequence for comparison with known sequences of cancer-causing proteins. For the actual observation, we propose to grow the colon cells in vitro in 3 test tubes and then add the 3 types of bacteria, E. coli, F-nucleatum, and ETBF, to study if and how proteins move from the bacteria to the colon cells. In order to be able to see the proteins, the gene that expresses green fluorescent protein will be added to the proteins of each bacteria. Using a fluorescent microscope, the fluorescent protein's localization patterns can be observed in order to determine whether or not the proteins of each of the 3 bacteria were secreted into the environment. The fluorescence of the proteins makes it easier to track if proteins were secreted from the bacteria and were able to bind to colon cells. But for confirmation, the cancer-causing fluorescent proteins can also be removed from the bacteria and isolated in vitro to determine if cancer growth will still occur. The proliferation of cancer cells in each of the test tubes containing bacteria can be graphed every 12 hours in order to have comparable data with the test tubes that only contain proteins. If the proteins are able to cause cancerous growth in colon cells without the presence of bacteria, it is clear that the proteins are the sole cause of colorectal cancer. If the proteins do not have any effect on the healthy colon cells, then it is reasonable to assume that the specific bacteria cause proliferation and the cancer growth. Finally, to create a solution, it would be reasonable to compare the path each bacteria's proteins take in order to determine if they are able to directly travel to the nucleus of colon cells or are transported into the body cells by vesicles.
There are many challenges that are opposed to our project. The first one is that proteins cannot be tested in vivo. In fact, with our instrument, they can only be tested in vitro. The second one is that it's difficult to study proteins with the microscopy techniques that we have already. The third one is that even if price is making even if price to make this experiment is reducing, it is extremely costly. The fourth one is that find what proteins can cause colon cancer is very time intensive because there are many proteins in every bacteria in our body, and for every protein to study, we have to study for 30 minutes every one.
All right, so overall, our project aims at finding whether proteins of bacteria cause cancer or if the bacteria itself causes cancer. So in the future, we could take some steps, such as combining different types of microscopy techniques, such as electron microscopy and atomic force microscopy, in order to better observe the protein and the gut bacteria. We could also develop an antibody in order to bind and destroy the cancer protein. And we could also create sort of a pill or a capsule that can destroy vesicles carrying cancer proteins by traveling through the digestive tract and binding to cancer-causing proteins, which would inhibit cancer growth. So this experiment is important because it would take us one step closer to finding a cure for colon cancer. It would help us to get a better understanding of the relationship between gut bacteria and colon cancer. It would lead to further discoveries of more protein found in bacteria, and it would also it could also help us to develop new methods and to prevent or treat colon cancer. So these are our references.
Okay, so I guess I'll start with the first question. So for your future directions, so you talk about using cryo-EM and AFM. Can you be more specific as to what you would use each of those techniques for in terms of what exactly you're looking at?
So cryo-EM and AFM would help us to get a better look of these proteins. These proteins inside these bacteria are extremely small, so it would help us to look at these live samples, and we could also stick them under there to see exactly what the growth is, and we can look at it and observe it over a long period of time.
So would you be trying to track like the location of the protein, how many proteins, what the protein is doing? Like what exactly is it about the protein that you're looking at with these techniques, I guess is what I'm asking.
We're trying to find the location of the protein, so the different types of microscopes would help us to better take a look at these very small proteins.
And also to add on, atomic force microscopy can be used to study like the replication of bacteria. So if some minor adjustments were made, it could be used to study even smaller proteins and see how they proliferate and how they grow when grown in vitro.
So you explained very well how you will use positive controls and negative controls in your experimental design, and how you will use fluorescence microscopy to follow either the protein or the bacteria. You also mentioned that you will use colon cells, right, in your assay. And I guess my question is, how would you measure that these cells become cancer cells? What is it that is going to tell you that what you are doing or adding to the culture is going to turn those cells into cancer cells?
Well,
and how would you measure it?
We can measure that by graphing how much the cancer cell or the normal colon cells, if they start to proliferate tremendously and they don't stop, then it's clear that they're becoming tumorous, and that indicates that there's cancer growth. So if we are able to record that data, it will show us how much growth is occurring and if it's linear growth or if it stops at some point.
You guys have done a great job. It's very impressive.
Thank you.
You mentioned that you want to use different kind of microscopy techniques to look at things. So just wonder, why do you want to look at them? How would that help you in this particular study?
Could you repeat the question real quick?
So you want to look at things, right?
Yes.
You guys, you're going to see things. So why is that important?
Oh, the importance of looking under the microscope would be with the fluorescent microscope, which we have tagged the proteins to become fluorescent. So we can track if the proteins in bacteria are excreted and bind to colon cells, or if they remain in the bacteria. And the bacteria will cause proliferated growth.
Yes, one more question.
So I also think this is a very great presentation. And as you guys mentioned, there's a time and a cost factor. One of the things I think you did really well was you went and researched where the current state of the art is. Not just found bacteria that were associated with it, but putative mechanisms, right? And you've selected these 3 proteins that you wanted to look at in particular, right? Then you also said you want to look at many, many other proteins, right? You want to scan if it's the proteins, all the proteins in the bacteria, for example. Except for the microscopy facilities we went to this week, do you guys think have you guys seen another facility that would be able to help with that kind of time-intensive process here at the CNSI?
Yes. You have asked a very intelligent question, and
I can short circuit it. It's just simply put, you guys went to the 6th floor and saw the screening facility.
Yes.
That's the type of facility that's dedicated to doing exactly the experiment you're talking about, where you've isolated all 100 different types of proteins. They can even grow proteins, each different protein in a well, suck them up, and test 100 proteins all at the same time in one batch, and that takes a half hour. And they can even do that proliferation assay you mentioned.
Yeah.
And so this idea of coupling what you can and images. So the point is, they've taken the technique. If you as the researchers go to this light microscopy lab downstairs and develop an assay, the technology is already there to support you to try to answer those, can we do it in time and can we afford the cost? And having all of this in one building is one of these ways we can leverage all these different aspects of technology to develop what you guys learn into something actually productive that we can then translate into these results you're looking for. So I think it's this very forward-looking and very realistic work that can be done in a place like this. So.
Yeah.
All right, so before you guys get up, so first of all, let's thank all of the first 5 groups that have gone. You guys all have done a great job so far. And since we've been sitting here now for about an hour and a half, we're going to take a quick 10-minute break to stretch, use the restroom. So let's meet back here in 10 minutes.
Harushi. No, I don't. Yeah, make sure to change.
Feet off the chairs, please.
Make sure to change it to UCLA Web.
All right, so looks like we have at least most of you guys in here. So yeah, so let's go ahead and get started. So you guys ready?
Yes.
All right.
Actually, you change this. Okay. Okay. So will you switch it on this? No. Okay. Hi everyone. My name is Arushi Madan, and I'm from Tesoro High School. Hi everyone. My name is Shreya Madan, and I also go to Tesoro High School.
My name is Araya Benavides, and I go to Downtown Magnets High School. And today we'll be covering solutions that can be designed by identifying misfolded proteins in patients with Parkinson's disease.
Parkinson's disease gradually disrupts a person's brain, which causes shaking, stiffness, difficulty with walking, balance, and coordination. Patients with Parkinson's also lack norepinephrine, which is a main chemical messenger of the synaptic nervous system. The protein structures of someone with Parkinson's are misfolded into amyloid fibrils proteins. Misfolding and inclusion body formations are common events in neurodegenerative diseases characterized by deposition of misfolded proteins inside or outside of the neurons. Our question relates to identifying the issue before it occurs. In this case, our goal with our proposal is to identify when the proteins in the human body start to misfold in order to design possible solutions such as an antibody to prevent this disorder. Techniques such as cryo-electron microscopy can provide 3D images of these protein structures. Previous research has shown that misfolded proteins form clumps of proteins known as Lewy bodies. A team of researchers at the University of Pennsylvania injected the misfolded alpha-synuclein into a part of the mouse brain rich in dopamine-producing cells. Lewy bodies began to form. Thus, Parkinson's disease began to develop in the mice. However, not much research has been done on the idea of identifying the misfolded protein beforehand and attempting to prevent it from misfolding. Currently, doctors are utilizing single photon emission computerized tomography scans to identify if a patient has Parkinson's disease. In terms of social context, Parkinson's disease affects 60,000 Americans each year and 10 million people globally. Generally, people start to develop Parkinson's at the age of 50 and above. It is rare to see development before this age. Many patients are suffering with this disease in their daily lives and cannot live normally even by themselves. In 2013, the Obama administration launched the Brain Research through Advancing Initiative Neurotechnologies Initiative, or also known as the Brain Initiative. This initiative is aimed to achieve the development and application of innovative technologies that can create a dynamic understanding of brain function. It began to one day cure brain disorders such as Parkinson's and Alzheimer's disease. Furthermore, researchers have established how a protein called alpha-synuclein, which is closely associated with Parkinson's disease, functions in healthy human brains. By showing how the protein works in healthy patients, the study offers important clues about what may be happening when people develop the disease itself. Parkinson's disease is one of a group of conditions known as protein misfolding diseases, because they are characterized by specific proteins becoming distorted and malfunctioning. These proteins then cluster into thread-like chains which are toxic to other cells. The researchers tested mutated forms of alpha-synuclein that are linked to Parkinson's disease. This was found to interfere with the same mechanism, essentially by impairing the ability of alpha-synuclein to regulate the flow of synaptic vesicles and hence compromising the signaling between neurons.
According to scientist Sarah S. and her team at the University of Basel in Switzerland, they are using light and electron microscopy on the brain tissue from 5 Parkinson's disease brain donors to identify the alpha-synuclein or protein structures. Also, scientists are able to use STED microscopes to confirm that the Lewy bodies consist of crowded structures and dysmorphic structures. This technique also allowed scientists to discover a protein-lipid compartmentalization. Scientists from Harvard University are currently taking skin cells and converting them into stem cells. Stem cells can be converted into any tissue in the body, including brain cells. Hence, with this technique, scientists can observe the Parkinson's and other neurodegenerative diseases because stem cell-derived neurons can help them study this disease. But even with that current research being done related to Parkinson's disease, we still do not have a cure.
We believe that using both the atomic force microscopy and cryo-electron microscopy simultaneously to take the image of the protein before degeneration and compare it with images of healthy protein structures in order to understand the differences between the two structures will be beneficial for curing this disease. Atomic force microscopy, also known as AFM, uses a probe that scans the surface of the object being studied and allows scientists to obtain the mechanical properties of the sample. Additionally, the cryo-electron microscope is an alternative approach to X-ray crystallography. This means this technique can scan macromolecular structures without the need of crystallization. Once the image is captured, then a high-resolution 3D model can be designed of the protein, similarly to what is shown on the screen. We believe that if these two methods are combined, possibly a cryo-EM AFM, then scientists could develop a highly advanced image of the proteins. The image would allow scientists to create a drug or an antibody that can be injected into the body before the proteins are misfolded and damaged to prevent these complications from occurring. Also, since the AFM has a tip, an antibody could be attached to the end of the tip and unfold the misfolded proteins, then possibly realign the proteins back together.
The challenge to the proposal is that once we freeze the protein sample during cryo-electron microscopy, the probe of the atomic force microscope will not be able to scan the frozen sample.
In order to resolve the problem, we propose to add heaters in the area where the protein is being scanned in order to melt the water. Then add antibodies to a substrate to make sure that the sample does not flow off with the water, as well as add antibodies to the probe in order to prevent the proteins from misaligning. In conclusion, in order to try a new perspective for the study of Parkinson's disease, our proposal suggests that the combination of the atomic force microscope and the cryo-electron microscope will be beneficial to capture a high-resolution image of the protein right before it starts to misfold. This will allow scientists to further enhance their understanding of the misfolded proteins that are responsible for Parkinson's disease and possibly create a drug or an antibody in the future to prevent the proteins from misfolding. Once a potential cure is created, scientists can test the drug on rats or any other form of animal that has a similar brain structure to humans and eventually introduce it to humans. This will hopefully cure Parkinson's disease. Thank you.
All right, questions?
So you said the proteins are misfolded. Would applying the antibodies unfold the proteins? Is that the path?
Yeah, so the atomic structure or atomic force microscope and the cryo-EM are hopefully our proposal takes a clearer picture of the protein. So then once we inject the antibody, whether it's to the protein or when we discover the structure, we can propose an antibody that will refold them back to normal.
Okay.
Students, do you guys have any questions?
So you're interested in sort of understanding the onset of this misfolding, right? What's the where do you plan to source the proteins from, right? Like, how do you plan to get proteins that are properly folded and misfolded for this set of experiments that you have, right? Because you need samples, right? Are these going to be purified samples? Or are you going to get them from patients? And then once you have these proteins, how do you
how do you get them over this time course, right? Is it just a matter of many, many, many experiments and hoping that you find one that's starting to misfold, or what's the approach there?
So we were, as we were researching, we found out that the decrease of dopamine levels actually cause Parkinson's disease. So whether we inject the lower levels of, or inject lower levels of dopamine levels in rats, let's say, then we could, their protein structures will automatically start to misfold because that's the leading cause of Parkinson's disease. So we could obtain the samples from rats or whatever organism we test the low levels of dopamine. Yeah. And then the time period. Oh, and can you repeat the second part of your question? Was that it, all of it? Oh, okay.
Okay, so you're talking about trying to combine the utility of the AFM and the electron microscope,
Yeah.
right? So the way you proposed it, there's a bit of a problem. First, what do you think is a potential problem with trying to heat up a sample that's frozen in an electron microscope?
Well, we ran into that problem. Like, if we freeze the protein structures, then we can't actually obtain the image of the protein itself because it'll be frozen and the tip will not be able to go through the frozen sample.
Right.
So if we heated the sample, then the water could flow off and we'd have actually a substrate with an antibody attached to it. So that way the actual sample of the protein can stick onto the substrate and not flow off with the water once it's heated and melted off.
I think that's a very interesting question, and whether the protein could stay there, that's a very interesting thing, that the protein could even be suspended in the vacuum, but where's that water going to go inside of that electron microscope? It's in a vacuum, it's going to evaporate, and those engineers are going to be very unhappy. On the other hand, if you do the AFM first off the fresh sample, then you could do it in your device if you built a device to freeze it in place.
Okay.
Right? So if you reverse the direction of those two, you actually could make it perfectly compatible, right?
Okay.
And the second thing I would ask is, if you're talking about putting an antibody on a tip of an AFM and then trying to capture a protein like the Lewy bodies, those type of proteins, what happens if you put that into a cell that doesn't have Parkinson's? Is that protein still there? And are you just going to get a bunch of loose protein on the tip? Are you pulling on like an individual protein, or are you planning on pulling on the body itself? Could you clarify that just a little bit?
We're planning to pull in the body.
Yeah. Yeah, because if we were to do it in like a healthy cell without Parkinson's, the proteins wouldn't be folded. So we are planning to do it on the body itself.
Thank you. Thank you.
Hello. Hello everyone. Today we're going to be discussing the topic of solving addiction with nanotechnology. My name is Harrison Lin from Portola High School.
I am Ishan Sharma from Miraloma High School. And I am Joshua Stevens from Walnut High School. So what technologies and nanotechnologies can scientists use to alleviate patients from drug addiction? Drug addiction has been a common issue for many years now, and the abuse of drug use has caused many detrimental, long-lasting, and permanent effects to an individual's health. Scientists have figured out ways to slow the process of people getting addicted by inserting siRNA or small interference RNA into the body of the test subject, later nullifying the DAPPR32 protein, which will be explained later on in more detail. Instead, with the use of two-proton excitation and confocal microscopy, we will investigate how inserting ibogaine will affect the DARPP32 levels. So, what actually is an addiction? Addiction is a complex condition, a brain disease that is manifested by compulsive substance use, despite the harmful consequences. For this presentation, many drugs have different side effects, but all cause the brain to secrete the well-known hormone which is also known as dopamine. And around 21 million people in the United States alone were addicted to substances such as nicotine, cannabis, etc. And there is a specific reason. In the brain perceives two systems as liking and wanting, and they view this as two separate psychological experiences. Liking refers to spontaneous delight, while wanting refers to a desire for more. In this case, certain drugs acts as the warning mechanism, which causes people to crave for even more drugs and decreases the tolerance level.
So let's talk about some history regarding addiction as a topic. So addiction was first surfaced as a disease since in the early 19th century, and the concept of addiction at first had many people doubting the legitimacy of it because there wasn't enough research following up on the claims made for this disease. However, there were some examples like the Opium War between China and Britain, which created a sense of realization that there's actually some danger regarding addiction. And finally, in 1997, the ASA, which is the AMA, which is the American Medical Association, and other medical organizations around the world, finally officially made this a disease. So our process is complicated but simple in a way. So we have this 3-step plan: examination, testing, and conclusion. So examination, I'm not going to go into great detail as we're going to be explaining in further detail in the further slides. But here we're just going to talk about examination is basically we're going to be creating some samples and testing out some. And then we're going to move on to testing out these samples. And then finally, we will be finalizing these results.
So how are current scientists approaching this problem? So we have a case study that shows how scientists tried to use gene silencing against DARRP32. What they did is they basically took siRNA, which is small interfering RNA, electrostatically complex to golden nanorods, which would be transferred to the targeted areas affected by DARRP32. So in this figure, the host staining is used to show the siRNA. So in figures A and C displays normal siRNA, while A shows the golden nanorod complex actually being successfully combined together to the siRNA. And figure B and D just shows regular DAN cells. So in this figure, we have a positive control, the new complex that they were experimenting with, and another positive control. Obviously, the negative control siRNA was expected to have no effect on reducing the amount of DARP32 present in a human. So obviously, there's no results there. As for the siRNA nanocomplex with GNR, it showed a 85% effective reduction rate in 24 hours, and 87% reduction rate in 96 hours, and I can't read that from here. What does it say? 81%. 81% reduction rate in 96 hours, or
1 week.
1 week, yeah. In 1 week. Meanwhile, our positive control is actually another product that's already been released that has similar effects to the new nanocomplex that these scientists are experimenting with. However, as you can notice, it is much less effective, and it takes more time to reduce the amount of DARP32 present in a human. And then these results show how much DARP is present in a human after 120 hours of being in a human body. So the negative control shows that there's no change whatsoever in the amount of DARP32 after 120 hours. Meanwhile, the new nanocomplex that the scientists were working on, it shows an effective 67% reduction rate of DARP32 in the human body. However, and then its competitor product, which is the positive control, only shows a 29.7% reduction rate.
So let's move on to the project proposal. So as we all know, previously stated that addiction is a relatively new disease relatively to other diseases out there. So the idea of the relationship of nanoscience and addiction is really new. So our group itself has developed our own hypothesis and solution that can, in theory, might be able to help treat addiction as a disease. So we will be using platelet cells in the blood. We'll be using platelet cells in the blood plasma. So blood plasma, as we all know, carries proteins and and is around our whole body. So a company called Platelet Biogenesis, so then they have this idea of using platelet cells inside these blood plasma and use transfusion as a way to spread this cure. So we're going to be inputting an alkaline called ibogaine. So ibogaine is something that rehabilitation patients use when from cocaine or heroin. So we will be using two photon microscopy, which is a relatively new microscopy compared to other techniques out there. And also the confocal microscopy to observe the individual platelets cells through the process of eliminating DAR-PP32. So jumping back to the platelet biogenesis, their idea was to, from human-induced pluripotent stem cells are poised to become among the first stem cell-derived tissues ready to clinical use, representing a major first step towards a sustainable donor-free blood system. So the hypothesis. Our hypothesis is we will genetically engineer platelet cells inside blood plasma with ibogaine, which will specifically target the DAR-PP32 and it will act as antibodies and eventually eliminate all secreted dopamine. And the microscopy imaging techniques. So we will be using two different microscopy techniques. The first one will be the confocal microscopy, as I mentioned before, and the two photons microscopy in the research. So the confocal microscopy will provide us 3D images regarding how the ibogaine is countering against the DAR-PP32. And the two photon microscopy is obviously it can look into deeper tissues and the DAR-PP32, so we can look at how the relationships has changed and if our hypothesis is correct. So some social context about this. So by doing this, we will develop methods to combat addiction and learn more about neurological disorders because in the science field we haven't fully understood the whole science behind the brain. And there will also be potential new cures for other diseases as well. So some future directions. The platelets can potentially target other kinds of diseases as well, not just addiction, because as we all know, the platelet cells can carry many different forms of treatment, not just ibogaine. And yeah.
Okay, so overall, the usage of platelets in blood plasma as a carrier for certain elements or proteins could be a solution to our problem of drug addiction. By using ibogaine to target DAR-PP32 in the brain, we can lower the levels of dopamine within the brain itself and decrease the occurrence of the patient of becoming a drug addict. We will use certain microscopic techniques as mentioned earlier, such as two-photon excitation microscopy to image the action occurring within the brain, allowing us to conduct our research. However, since this is a viable solution for solving just addiction, we can theorize that we can use the same theory, not just to solve addiction, but for more elements in the future. We can use the same procedure to target different parts of the body and certain diseases which we could not yet cure and will be investigating in the future. And the Brain Initiative seeks to understand the basis of our own brain, and by using technology and nanotechnology to seek these sites of the brain. The Brain Initiative has been successful using nanotechnology and technologies such as certain microscopes in investigating common brain diseases, and now we have understood more of the brain overall.
So the contribution, just like Ishan said, this will be able to help cure other diseases as well, potentially. And since we have such a limited information regarding nanotechnology and the idea of addiction, we will be able to do more research and eventually have more info regarding this. and some challenges. So we will be facing some challenges. As we all know, addiction is in living samples, so like humans, and we can also, but at first we're gonna be testing out these examples in like mice or other animals out there, and because we cannot input this technique onto a human without actually testing onto it first. And also delivering light to the brain. As we all know, microscopy, we use several different techniques, and we usually input light into the brain so we can look carefully. And these are the references. Thank you.
Okay, so I've got a question. Are you guys worried about the effects of drastically decreasing the amount of dopamine in one system, like depressive effects?
So you're saying if it's gonna be over killing the dopamine?
Yeah.
So so based on our research, the blood plasma cells, we can control how much we input into the platelets. So then we can analyze how much the dopamine is over the normal amount, so we can control the amount how much we put in, so that we would notify it, basically.
So along the same lines, you explain how you were about to you will use platelets as a carrier. How do you envision the delivery? How would you deliver your drug in the place where you wanted it to be delivered, rather than having it everywhere in the body?
So as we just said, we're going to use transfusion as a way to get into the body. So right now we're still discovering, in our research, we're still discovering different ways for us to input this into the body. So some ways is that there's so we can genetically engineering them so that they'll be staying in the neural side of our human body instead of going into like other areas of our body by genetically engineering them and changing their structures.
Gentlemen, very bold initiative. Obviously, something that affects many, many people. To answer some of the question, I guess if you were really wanting to look at something that affects the dopamine areas, perhaps instead of doing transfusion, something like something that's directly injected into the CNS space through like a lumbar puncture or something of that nature. But my two questions relate to the confocal microscopy. When you use that and you inject ibogaine or the DARP32, does it look different under the confocal? Those that are affected with the DARP32 or the ibogaine, do the platelets look different under confocal microscopy?
So on the case study that we showed the scientists that did the case study, they used siRNAs to notify the DARPP32, and we are using the blood plasma cells. So if we look back on to the charts that we showed, we can see that after the siRNAs were in contact with the DARPP32, their structures were broken down from the siRNAs. So we can obviously we have our future directions. We have to be testing this, so we can hypothesize and theorize that it can have the same effect, because ibogaine is basically slowing down and eventually not curing, but slowly treat the patients off the temptation of drugs. So we can theorize that the structures will look the same as the siRNA. Yeah.
I guess it relates to the question was, what are the unintended consequences of suddenly dropping, inhibiting that? Are there, other than dopamine, are there other effects that you find anything when you were researching or reading about that? Are any other effects of this particular drug, ibogaine?
I don't, not as we know of in.
I think, in my opinion, basically what dopamine does, it basically triggers like your level of happiness, and basically dropping the levels of dopamine too far will probably lead you into depression. And maybe if you're like in a depressive state, it might cause the person who was already addicted to drugs to take even more drugs. So we have to be very specific with our measurements in order for this to actually work.
So just like Sean said, that's why we're gonna genetically engineer them into the right amount needed.
Thanks. Very, very interesting.
Well, if that's all the questions, then thank you guys. All right, next team. Team 8.
Hi, I'm Andy Fitzgerald.
I'm Kaylee Ching.
I'm Coco.
And we are here to present our proposition for the application of quantum dots in the brain.
Okay, so throughout history, there have been a few factors about your life that have been deemed unchangeable. One of these is your height, which is it's primarily determined by your genetics, so you can't change it. Just recently, we've been coming up with ways to create human growth hormone in the lab, but that still has problems. So through this method that we are proposing, we have shown that we can stimulate the hypothalamus in a human brain to create more growth hormone in order to increase one's height. Because I gotta say, some of us just get the short end of the stick. So, okay, yeah. So we propose to test this in vivo on a rat model first. So it is hoped that this novel technique will begin to elucidate the mysteries of the brain, as well as pave the way for an eventual human model of this technology. Because of the way this technique is set up, a human model is not quite, the science is not quite there yet for a human model.
So our question is, can nanotechnology and nanoscale microscopy be used to artificially stimulate brain neurons? So background of LED and quantum dots. LEDs are used in many applications in life. They are commonly made of yellow and red phosphors, but the problem with LEDs is that they have wide emission bandwidth, especially for red phosphors, because the red color is on the edge of the sensitivity spectrum of our eyes. So to overcome this problem, quantum dots have been developed. They are made of new phosphors and phosphor combinations, which are green, yellow, and red. This discovery provides a higher quality of light than LEDs. So quantum dots are semiconductor light-emitting nanocrystals with nanometer-sized structures of unique optical properties. They have bright fluorescence, which are able to emit fluorescent light of different wavelengths. They can also be used as nanoscopic light bulbs to light up and color specific cells that need to be studied and under a microscope. These properties make quantum dots a perfect tool for visualization of brain structures and the mechanisms underlying its function. Due to their unique properties, even single molecules can be observed. And this image shows the blue LEDs excite the red and green cadmium-free quantum dots, resulting in high-quality white light.
Now you might be wondering, how do these quantum dots glow? Well, for that we need to go down into the quantum scale and look at the single atoms. So when the single atoms are excited by a laser, what happens is that the single atom loses an electron to the actual orbital surrounding the quantum dot because it functions like an atom itself. So it loses an electron to the gap where an electron should be, and the electron on the outside of the orbital of the quantum dot, they bond together. And when the electron finally becomes, and that's called an exciton or exciton, not sure how to pronounce it, but when that electron comes back into the original atom it came from, that's when it produces that fluorescent glow that they're known for. Now you might be thinking these are really expensive and hard to create, and I can tell you from what I know, they are not. They are easy. Well, some of them are easy to create and cost effective, like this week we've been trying to create them in our dorm. Also, one benefit of these quantum dots is that they offer less photo bleaching and last for a very long time compared to other types of fluorescent microscopy, like GFP. Okay, now, fluorescence microscopy. To understand fluorescence microscopy, you have to understand fluorescence. Fluorescence is the emission of light. Now, and fluorescence microscopy requires fluorophores, or something that glows, and the main one used for this is GFP, but we will be using quantum dots to accomplish this. Okay, and another main topic in fluorescence microscopy is the red shift, where a light that shines, if you shine a blue light at a quantum dot, a green light will glow back. It shifts the color one color towards red on the spectrum. Another reason why we chose quantum dots for this is because of the localized imaging. Quantum dots can deliver light to very specific areas of like the brain, whereas if you use sound or other types of light, you're not going to get that precision that a quantum dot offers. Okay, now our plan. Let's see. Okay. Our plan. So for our plan, we need to first get our materials. Our materials require quantum dots of certain size. Carbon quantum dots would work. And we also need genetically engineered rats with a photosensitivity to a certain bandwidth of light in the brain. Now, from there, what we need to do is cut a hole, or like a little sunroof, in the head of the rat, which is why we can't really do this on humans. But this is so we can put the quantum dots in the brain and position them exactly where we want. Now this is where when the fluorescence microscopy comes in. From there, we're going to look with our little microscope and make sure those quantum dots are in the right place so that we only stimulate the hypothalamus and pituitary gland, because if we don't do that, we can have a lot of other issues. And the fact that you can actually find the specific part of the hypothalamus that produces human growth hormone has been proven by Akira Matsuno. Now, from there, we are going to take that genetically engineered mouse with a specific bandwidth of light sensitivity in the brain. We are going to shine a different color light onto its brain, and then from there, the red shift will counteract it, so only the hypothalamus, and actually I have it up here, only the hypothalamus and pituitary glands will be stimulated. The rest of it will just be fine. There'll be no other effects, in theory. And then from there, the brain will continue to produce more growth hormone, causing this rat to grow very large, or will cause it to grow much longer and much larger than it normally would. Now you might be wondering where we came up with this idea. Now people who are in the program, you might have seen this video before. It's this video of this rat, which it's use of optogenetics. So they're doing something similar that we are. They're also shining lasers in the brain, but this is so they can use light-activated predator instincts in the brain of that mouse. Now another application of this is quantum dots are currently being worked on for use in the human brain as a cure for Alzheimer's and Parkinson's, to stimulate other parts of the brain.
Now we hope that with our research, other scientists can perhaps add on, because there are some aspects of our research that still remains unanswered. For example, it would be ideal if we could find a non-invasive way to get the quantum dots into the brain, as well as getting a light source, because the current method we propose, we would have to have, as Andy described, a sunroof in the rat's brain. So if we could solve and answer these questions, we could perhaps apply this to humans to cure brain disease, and also there's the possibility of curing the growth hormone disorder.
Okay, So, possible solution to GHD of human. GHD is growth hormone deficiency, which occurs when the pituitary gland doesn't produce enough growth hormone. It affects children more commonly than adults. So, GHD can be present from birth, resulting from genetic mutations or structural defects in the brain. It can also be acquired later in life as a result of infection, radiation therapy, or tumor growth within the brain. So there are two ways that we can insert quantum dots into the human brain as a possible solution of GHD. We can use carbon dots or traditional quantum dots with biocompatible molecules. So the difference between them is that the carbon dots can penetrate blood-brain barrier without conjugation. In an experiment done in 2015, the data indicates that the carbon dots are able to freely penetrate the blood-brain barrier and precisely target on a specific tissue. So similarly, the carbon dots can also possibly target the pituitary gland precisely. In contrast, the quantum dots are required to conjugate with a cell-penetrating peptide TAT in order to cross the blood-brain barrier based on the fact that quantum dot itself cannot label the brain tissue. But the quantum dots with biocompatible molecules, which is the peptide TAT, can also be used to target on the pituitary gland as a possible solution of GHD.
So we also hope that our research can contribute more to science by finding out more uses for quantum dots because they're a relatively new technology. We also hope that we will provide more information for the stimulation of hormones using quantum dots.
And just to add on to that, another contribution is using quantum dots to stimulate the brain. You could stimulate all parts of the brain, causing all other reactions. You can release all different types of hormones. This could be widely used if we could only find out how to not cut a hole in someone's brain. And this brings us back to the original question we asked at the beginning. Can nanotechnology and nanoscale microscopy be used to artificially stimulate brain neurons? And through the methods that we have stated here, we think this is possible in a rat model. And
these are the resources we used. Thank you. Any questions?
Oh, dang. We're going to get a lot of questions. Okay. You. Okay. Or we'll hand you
Hi. I have a question about the human growth hormone. And
Yeah.
I was wondering, like, you say that you could use this to, like, increase people's heights.
Yeah.
Are there other things other than just human growth hormone that contribute to this?
Well, okay, but human growth hormone is the one that increases the main height, so it can increase, like, basically, it mainly increases, like, muscle growth and, like, long bone growth. So they actually do use human growth hormone that's lab produced in normal people. But this is just a way for you to use your own human growth hormone that your brain produces that would be much more natural.
Okay, I have a follow-up also.
Okay.
For the different, like, ways, for the different, like, causes of the disease, you talk about, like, I think damage to that part of the brain. How would this be used if, like, that part is damaged?
You want to take that one, Kilton?
Or not developed properly?
I'm not sure about this part because we are not mainly focused on the GHD. We are just proposing like a future experiment that we can be done by scientists through our experiment on rat model.
Okay. Well, also one thing I'm going to add is if your pituitary gland is like missing or like not there, then I don't think this is the right treatment for you. I think there's going to be other ways for you to solve this, and this can only help you if you have at least a semi-functioning pituitary gland and hypothalamus that can actually create human growth hormone.
Okay, thank you.
You're welcome.
There was a couple of questions.
We got a lot of questions. I don't know if that's good or bad.
That is good. Have people's attention.
Yes, we
If
do.
you were to put a ballpark estimate on the probability of screwing this up irreparably, like permanently screwing up somebody's brain, what would that be?
Well, I mean, first of all, we're not going to jump first to the human model because no one wants to have a hole in their brain just so they can grow 3 inches taller. So I think first we're going to do a rat model, but if we could figure out a way, I was thinking maybe like nanobots. If a nanobot could like crawl through your nose or something, deposit like your quantum dots, and then leave, that wouldn't cause that much damage because these quantum dots are tiny. Basically, they're one 100 trillionth of the size of a single brain cell. So if you could find a non-invasive way to get into the brain, having these quantum dots in there would not be a problem, at least that I know. Oh, we got more questions.
So you guys talked about like artificially changing, like the possibility of artificially changing the height of a person.
Yes.
Do you think ethical questions will be raised like against this?
Okay, well, people are already doing this. This is already a technique. Not this. The quantum dot thing is completely new, but like the idea of changing someone's height, adding more human growth hormone to make them grow taller, this is already being used. And one thing that I just want to add is a lot of the times they use human growth hormone, like it's technically not the approved use of human growth hormone. The only approved use of human growth hormone is if you have a growth hormone deficiency, but people often use growth hormone to just grow taller just for like purely aesthetic reasons, and that could be damaging too because growth, like artificially lab created growth hormone does have some bad side effects that this might not. Oh, we got it.
Hi. I thought it was a great presentation. It was really interesting to learn about. Okay, so I'm just a little confused on, I understand that the quantum dots can like target the pituitary, or you can put them in the pituitary gland. How does, because they're phosphorescent, right?
Mm-hmm.
But how do they like lead to the pituitary gland actually secreting growth hormones?
Okay, so first you'd have to put them on the pituitary, the section of the pituitary gland that secretes and creates growth hormone. From there, you just stimulate the brain into and kind of trick it into thinking there's a lack of that, which has been done in before studies.
Okay, thank you.
You're welcome.
Yeah, and adding to that, I believe when you shine the light on the quantum dots, they like emit energy.
Right.
Yeah. So. Dr. Bentioli, I think you had a question?
Sure. I got a lot of questions.
Okay, I don't know.
I just wanted to say that's exactly why I love this class every year, because I get to learn something new. And I say that as a compliment because until 3 days ago, I even didn't know we could make quantum dots out of orange juice.
Yeah, you can.
And just by you bringing up that subject, I mean, that led to a discussion, and I did learn something. So it was
Yeah,
pretty cool.
this is a side note, but for like the last 2 days, we've been spending like an hour in our dorm, like our dorm microwave, trying to make quantum dots in the microwave, and it might have worked, it might have not, but we've been trying. Yeah, it's being imaged right now. So we're using $5 quantum dots in a multi-million-dollar machine.
So just, we can wrap it up because this is running, but just to your question, if you're there's a lot of interest in this, obviously, is one, they mentioned a word, optogenetics. Okay, it's a very hot area, which is using light to basically modulate the activity of your genetic system, right? To target proteins that can become light sensitive, or genes to become light sensitive to produce proteins. So that is most likely the mechanism by which this approach would take. So if you're interested in our ability to control cells in your body using light, it's a new field called optogenetics. If you want to keep thinking about this, to get rid of your sunroof in the brain, there's another type of nanoparticle that's quite new called an upconversion nanoparticle. And what that does is it takes near-infrared light and converts it up into blue light, right? And infrared light can actually penetrate through tissue, right? So then you don't have to necessarily, there's a very recent work trying to do that so that they don't have to have the window in the brain. Infrared light has far deeper penetration, and if it's incident then on what's called an upconversion nanoparticle, it can produce the blue light required to genetically stimulate through optogenetics. So there's a lot of work in this area that's quite new, but you may see it, you may not. We certainly don't want sunroofs in our brain.
I could see that could be problematic. Thank you.
Thank you.
Addressing the Effects of Broad Antibiotics on Mutualistic Bacteria by Taryn Sager,
Christina Sanchez,
and Lydia Kim.
In the sense of broad spectrum antibiotics eliminating both mutualistic bacteria and pathogens, which cause disruptions in the microbiome and dysbiosis, what can R-type pyosins do to address these challenges?
The following terms will be brought up several times throughout the presentation. R-type pyosin is a bacteriophage with contractile bodies that comes from Pseudomonas aeruginosa bacterial strains, while a broad antibiotic is an antibiotic that acts upon a wide range of pathogenic bacteria. And the technical good bacteria are bacteria that are involved in a mutualistic relationship with the host, with an example being gut bacteria. And finally, dysbiosis, which is an illness in which there is an unbalanced amount of harmless bacteria in the gastrointestinal tract.
Our abstract. So today we'll be talking about R-type bacteriophages. So essentially the problem is that broad-spectrum antibiotics are targeting all types of bacteria, including good, and we don't want that. So to combat this, precision antibiotics were created to target specific bad bacteria, and R-type pyosin is one of those. But its killing range is really limited to only a few types of bacteria. So what we want to do is expand that killing range, but still keep it contained within that realm of killing only bad bacteria and not the good. And we can do this by swapping out or fusing tail fibers of the tail fiber of the pyosin with tail fibers of other phages. And what this does is that it changes the bactericidal specificity of the R-type pyosin to that of the phage that it swapped tails with. So the concept/topic. There are two sort of topics that we'll be mentioning today. First is the R-type pyosin, and next will be the scanning electron microscope. So for the R-type pyosin, if you see in the picture, part A and part B, part A is a scanning electron microscopy image of the R2 pyosin, which is negatively stained with phosphotungistic acid, and part B is a diagram showing the major structures and parts of it. And in the scanning electron microscope, that image is just a diagram showing the parts and just labeling it.
Precedence. In this day of age, broad-spectrum antibiotics are used as the last resort for bacterial infections. However, the effects of taking these broad-spectrum antibiotics include antibiotic resistance and C. difficile infections in the colon, both of which are deadly. Pyosinase was the first antibiotic prescribed in hospitals, and just like R-type pyosins, pyosinase was also derived from P. aeruginosa, which was used, but consequently it was toxic and taken down.
Discussion. The first R-type pyosin was discovered in 1964 by Kageyama from the Pseudomonas aeruginosa strain. It's a bacterial strain. And the R-type pyosin has a rod-like structure with a double hollow cylinder and 6 tail fibers. What I mean by the double hollow cylinder is that it has a sheath and a core, and the sheath is contractible so that the core can penetrate the bacterial membrane to kill the bacteria. It also has a single hit mechanism, and it can kill bacteria with a single hit. And finally, the pyosin R1 and the T-even phage tail have been proven to have close morphological resemblance, and the T-even phage is also known as the E. coli phage. Contributions. The R-type pyosins have proven to be effective in killing harmful bacterial cells, and to support this, we have a supporting piece of evidence. In many cases, a single R-type bacteriosin is sufficient to kill a bacterial cell, and their specificity to target only harmful bacteria helps address the issue of the broad-spectrum antibiotics killing the good ones. And to back this up, although P. aeruginosa R-type pyosins kill mainly strains of P. aeruginosa, they also have been shown to kill some strains of Haemophilus nyrissa and Campylobacter species.
Limitations. R-type pyosins are a one-time use, and after each infection, the patient would have to receive a new dose. Broad antibiotics kill good and bad bacteria, while R types kill specific bacteria, yet their kill is limited to the specific pathogens as mentioned beforehand. This is good in a sense that they kill the specific bacteria, but we would like to see we would like to see R-type pyosins kill more. This was not enough. Unfortunately, there's no study in which a large number of pathogenic bacteria is killed without the good bacterial being altered. R-type pyosins are injected and can only be operated if the infected area is known.
A solution would be to essentially create a more precise version of a spread antibiotic, an antibiotic that won't attack the helper bacteria, but that settled within the body, but attack a larger range of negative bacteria. We could, we could use electron microscopes as well in order to give a more visual representation to check, to see the damage it would cost towards the good and bad bacterial cells. By replacing the pyosin tail fibers with tail fibers of Pseudomonas phage PS17, we changed the bactericidal specificity of ART2 pyosin particles to a different subset of P. aeruginosa strains, including some resistant to PS17 phage. We further extended this idea by fusing parts of R2 tail fibers with parts of tail fibers from phages that infect another bacteria, including E. coli and Yersinia pestis, and changing the killing spectrum of pyosins from P. aeruginosa to the bacterial genus, species, or strain that serves as a host for the donor phage. After finding a compatible phage for the R-type pyosin that will fuse or replace a tail, we could put it into a lab testing, which we would apply the R types to good and bad bacterial cells through a selection of wells in order to test efficiently on how badly or how well it affects the cells.
So after doing the lab, we can use electron microscopy to image the types of bacteria that the reprogrammed R-type pyosin attacked. So we could use scanning electron microscopes or transmission electron microscopes, and from the images, we can track the progress of the bacteriophage and see the damage it caused to the bad bacteria, and see what needs to be changed. If it attacked the good bacteria, the tail fiber that is implemented in it is obviously not working out. So from the images, we can just only improve from that.
Thank you for your time. Any questions?
So I definitely understood your goal of trying to expand the breadth of the targeting range. So, but it's unclear exactly how far. Are you trying to target it from like one species to 3 different ones? Are you trying to, find multiple mechanisms that can broaden, multiple different proteins and assess how they all affect a broadened, set of bacteria? Or are you trying to specifically switch the bacterial types, like just show that you can move it from one bacteria to a specifically bad bacterium that you have in mind?
So we're trying to broaden the range between the bacterial species that they can use, and by finding one of the tail fibers that would work for all of them, and only killing the bad bacteria, and we can fuse more than one tail fiber, so we can just find the specific per multiple disease, hopefully more than 10 at a time, and you're able to fuse them to create basically a super R-type pyosin.
Oh, to add on to that, essentially our process would be with R-type pyosin, they have tail fibers, and we can only switch the tail fibers of the R-type pyosin with another phage. We had a study that Christina read where scientists were actually able to switch the tail fibers of another phage with the R-type hyacinth. And what that phage was able to infect, the R-type hyacinth, when it was switched, when the tail fibers were switched, they were also able to infect that bacteria. So I guess our main goal is to expand the specificity that the R-type can infect. And I guess our way of like hand-picking which specific bacteria would be infected by the R-type hyacinth is just our method of doing so.
So you saw from Professor Miller's talk, right, that a cryo-electron microscopy was used in this case to understand the mechanism of action of these pyosins, that contractile nanotube functionality, right? I guess my question here is what do you hope to gain from electron microscopy specifically, and why you chose electron microscopy as opposed to an optical method, if your interest is to understand if these modified pyosins are killing cells, right? If they're being effective in your goal, right? Like, what is it about EM that you would learn? Because we know the mechanism of action and we know the structure, right? But EM is not giving you time course data. It doesn't give you a lot of information about whether cells are alive or dead, because they're already dead, because they're frozen, right? Like, what's the motivation for EM versus looking at optics, for example?
I guess the reason we chose EM would be that we did see that EM was able to identify the bacteria, the parts of the bacteria, and the interior of it, and that was just what stood out to us, that we could see more on the inside of the bacteria and what happened along with it. And it's also known that EM has a very, very high resolution that we can use to have a greater magnified view of the bacteria. That's just what was the defining point that we chose EM.
We would want to see closer and how in depth and see the different changes and how much affects each bacteria.
What's your plan if the bacteria you're targeting develops resistance to pyosin?
Okay. So we had a study that where R-type pyosin, we can constantly be switching out tails that will be able to target okay, sorry. What makes R-type pyosin, the switching tail fibers, unique is that it has a combination of that phage that it's switched with along with the R-type pyosin strength, and so that makes this unique combination of killing, which I don't believe that bacteria will grow to be immune to that quickly. But if they do, there is that possibility of constantly switching out tail fibers so that there's more diversity and less susceptibility to grow immune to it.
So is this technique limited by the number of phage tail fibers available?
Technically, yes, but it also depends. It depends on how many will react well with the R-type, but it also depends on how many will actually attack the good cells and if it will cause as much damage to the good cells that would be considered fatal.
Ladies, good forward thinking on this. Some of the unique things I'm thinking about this was that it's actually a good thing to have very narrow coverage. Most of the antimicrobials that we've ever had are always, even that they're called narrow coverage, are actually too broad. So this is extremely narrow. So identifying the bugs that you are trying to attack is actually a good thing, because it potentially looks at just one particular bug. My question is, how do you know when the phages switch out? What technique did you? Was the electron microscopy the way that you knew that there was switch? The different phages were switched. Is that how you knew?
That was, yes, you're correct. That's what we, according to our research, that's how that's how we would be able to see the different phage types.
So that was the reason for you choosing the electron microscopy technique rather than an optical technique.
Yeah.
All right. Well, then, thank you, ladies.
Thank you.
All right, last but not least.
Hi, I'm Nathan Lee.
I'm Alan Lowe.
I'm Gavin Seiden.
And we've done a thing on sugar-free sweeteners and the microbiome. No, it's this. Okay, so sugar is found in a lot of things that we eat, like desserts, candies, and drinks. But sugar is not very good for you because it causes blood sugar increases, which can lead to obesity, diabetes, fatty liver, and other bad things. So a lot of people will choose for like a light or like sugar-free alternative. And then the problem is that these artificial sweeteners usually have like side effects that affect your gastrointestinal microbiome. For example, maltitol, found in like sugar-free gummy bears, causes stomach pain and gas. So our study is going to find a solution to the side effects of like maltitol. All right, so the microbiome, before you have sugar-free sweeteners in the stomach, has a lot of bacteria, mainly from 5 phyla, Firmicutes, Actinobacteria, Bacteroidetes, Fusobacteria, and Proteobacteria. For example, lactobacilli converts nitrates into nitrites, and it helps prevent like pathogenic yeast growth and eczema. It's like it makes cytokines to inhibit skin inflammation and bacteriocins to kill other bacteria. So that's like supposed to be there. It's good for you. And then in your intestine, you've got thousands of bacteria, mainly from 2 genera, like the one on the left, the one on the right. And then the ratio of like the bacteria you have is caused by like your diet. For example, if you eat a lot of whole grains, you're going to have more Prevotella because they help you break down fiber and starch. Another type of bacteria is Bifidobacteria. You find more of it in breastfed babies because it helps them break down the human milk oligosaccharides and helps them make stronger gut mucus. And they live in the stomach, gastric mucosa, and the intestines. They live on the outer layer of mucus on top of the denser one that's on top of the epithelium.
Okay, so based on a study we found that was conducted on mice, the researchers were able to determine that levels of Bifidobacteria, lactobacilli, and SCFAs, or short-chain fatty acids, were increased in mice that replaced their diet from sucrose to sucrose being natural sugar, and they replaced the sucrose with maltitol. And the effect on the microbiome was generally good because fecal bifidobacteria is very common. It's very beneficial. It protects your gut from infections. Lactobacilli is also very common, and it's used in a lot of probiotics. Although it's important to note that having too much of any one thing in your system can be bad for you. So there was also heightened levels of SCFAs, or short-chain fatty acids, and which is pretty much the only truly worrisome result because SCFAs may decrease risk of infection, parasites, and cancer, but they also like generally contribute to diabetes and obesity, which are 2 very common ailments today. So this may show that the problem may not originate solely within the microbiome or changes to the microbiome.
In a study done in Germany in 1990, researchers radioactively tagged maltitol and fed it to both conventional and germ-free mice. In germ-free mice, in the excrement of germ-free mice, there was 39% radioactivity, and there was 59% radioactivity in the air. In conventional mice, the excrement contained 20% radioactivity and 72 to 74% in the air. The germs account for the 19% difference in excrement and the difference in the air as well. The takeaway is that the germs assist in the digestion of maltitol. The following are not related to the aforementioned study. Maltitol is broken down by bacteria using the enzyme maltase, hydrolyzed into maltose and sorbitol. The maltose is broken down by a bacteria into glucose, which is digested into carbon dioxide and water, which leaves through excrement. And sorbitol is also digested, which also produces carbon dioxide and water. For each mole of maltose, up to 12 moles of water and 12 moles of carbon dioxide is retrieved, and the water causes the diarrhea, and CO2 causes gas.
All right, so what we plan to study is, first of all, we're going to take the most common gut bacteria, we're going to put them on a petri dish, then we're going to expose them to maltitol, and then we're going to monitor the amount of CO2 they produce, because that will show us how much of the maltitol is being digested. And then we'll take the bacteria that do the most digestion, and then we'll attach fluorescent proteins to their maltase enzymes, and then we'll give them maltitol that is radioactively tagged with carbon-11, and then we're going to use fluorescence microscopy, STED microscopy, to watch as the maltitol enters the bacteria and then gets broken down. And then ultimately we're going to try and figure out whether we should prevent maltitol from entering the bacteria, or if we could just disable maltase, because you don't have to digest something to taste its sweetness. It can just pass through you undigested, and then you wouldn't have any side effects. So that's our goal. So you can eat sugar-free gummy bears without having diarrhea. And these are our references. Okay, questions? There is no slide after this. Just stretching. Okay.
Can you guys, so the last slide, can you go back?
Oh.
We just dig a little bit into what you plan to do, right? So you went through that. There's a number of different techniques, right? So what exactly what's the sequence again? Can you go through that again? And then let us sort of describe in greater detail when you're looking at these bacteria, right, that have maltitol in their environment. What do you hope to see from the optical microscopy that you're utilizing?
We want to find out we want to find out first which bacteria are mostly responsible for maltitol being digested, because there's thousands of them. You can't test all of them. And then so with the fluorescence microscopy, we're gonna attach fluorescent proteins to their maltase enzymes, and then it's kind of complicated, but we were hoping to use carbon-11 to attach the maltitol, and then use positron emission tomography to watch as that would go inside the bacterial cell, and then we could see with the STD microscopy it being broken down. So then we could figure out a way to stop maltitol from going through the membrane, so then it would never get digested at all. Okay, epic.
I'm just thinking about the complexities of that experiment.
So I think that's it's kind of ambitious to think. I think one thing you have to consider is radio labeling does not have any kind of high resolution, right? So labeling will give you total gross measurements from the whole dish, right?
Oh.
But what you're doing is actually possible. It's tricky, right? But I'll give you two extra techniques we haven't actually covered this week, but it's a good opportunity to bring this up. You have one more bit of what we can do with light imaging technology. is, for example, in that whole field of diverse bacteria, you have a challenge. You wanted to identify the ones that are actually doing the most metabolism, for example, right? You want to identify the ones that are uptaking the product the most. So maybe if you had, labeled maltose or labeled byproducts that you could look at optically and identify which ones were consuming the most, you could then actually cut those bacteria out, and you could sequence just those bacteria. That would allow you to identify exactly which bacteria were the culprits from a big mix of them, right? And that is something we do, is laser microdissection. You can actually use a laser to cut out just that one square where you think the offenders are, right? I think you have to think a little bit more about how you make the signal of who's producing the most maltose. Maybe you need the byproduct of maltose digestion to be fluorescent. So those bacteria that are making the most of those short fatty acids are the ones that light up. Things like that, right? So there's a couple practical details. But other than that, it is possible to accomplish what you want in there. And then if you want to use an optical method to monitor the enzymes and look at whether, for example, the maltose, the enzymes could be on the surface, they could be secreted, or they could be internal to the bacteria. And that could be helpful for understanding the structural mechanisms. Again, you could study this with light, and you could understand exactly where you'd want to try to apply a block to the bacteria, right? Do you want to get rid of the bacteria? Do you want to just block their product, et cetera, et cetera? So I think you've opened up a very good discussion about how you could attack these problems, and you've also connected it to a long-range health impact, which is that these byproducts actually have significant metabolic impact on people. And adding up all the sum total of all the different metabolic byproducts, having something that doesn't get digested as much could contribute to people not ending up with a long-term condition like diabetes. So I think being able to connect the basic grind of mechanistic understanding to long-term health impacts in a population is actually a very important product of science.
Okay. Sounds hard.
Any other questions from anyone? If not, then let's thank our speakers. And I just have to make sort of a general note that, I've been involved with this particular program. I think this is my 3rd year now. And every year I'm continually re-impressed by what you guys are able to put together in a very, very short period of time. You guys all did a really, really good job. So you should all give yourselves a nice round of applause for your work. And so just a couple of concluding notes. So the live stream is going to be archived on YouTube. So the same link that you use to watch the videos, if you're streaming, you can go back and watch yourselves present as much as you want, because it'll be online. Similarly, the photos that we took with you guys outside, we will be sending a link to everyone in probably the coming week or so you guys can have the photos that we took just out here, and probably a link to the slideshow that we're playing outside as well, so you can have photos. And then finally, as sort of one last parting gift for you guys, we have Nanoscale Microscopy t-shirts for this year. So we have a nice reception for everyone. You guys can go out, relax, have some food and some beverages, and we have the t-shirts organized outside by size. So students, please go and grab a t-shirt. And thank you guys all so much.